Can you see and can you also hear me? I would only like to know whether you can see and hear me. Is it visible and am I audible? Yes, please. We'll wait for a moment if someone is joining. Oh, yes. Thank you. Thank you. Yes, we'll wait just for a moment if anyone is joining. Yes, I hope like whomsoever I was expecting has joined. Can you please tell your names who have joined? I could see Ross, I could see Manasvi. Anyone else there? If I could know who has joined. We'll just wait for a moment and start. Yes, so now without wasting much time, because I personally have less time today, I don't know, there is something problem with my mobile's orientation. Today you have to adjust like this only. I'm so sorry. So anyways, the first question there, hardly 15-20 minutes, give me answers quickly so that we can, I'll just get my wife's mobile. Dikha to? You're calling him. वो होने के बाद दे दो. Right, the first question out there, the amino group transfer. I told yesterday, the amino group transfer, which vitamin is involved in the transfer of amino groups? Anyone out there, you know, which vitamin is involved in the transfer of amino group? The riboflavin, the pantothenate, the folate, or the pyridoxine? Which of these is responsible for amino group transfer, right? Riboflavin, I think it forms FAD, right? And FAD is involved in transfer of hydrogen. Pantothenate is a part of coenzyme A. Yesterday only I told, and that is involved in transfer of acetyl groups. Folate is a part of that folic acid. Again, like you can say is the coenzyme form of folic acid itself is riboflavin. What remains is your pyridoxine. So pyridoxine, it acts as a carrier for your amino group. Yes, yes, Ross, it is pyridoxine. I request others also to be awake. What fraction of Vmax is achieved with S of 100 km, right? We know that when your S is like when you say that Vmax is half of it, like when your velocity is half of Vmax, when V0 is half of Vmax, that time your S is equal to Km. So many people think that if we double the substrate concentration, then the Km will be reach Vmax. But we know that Vmax is not reached like that. Mathematically calculating, if you see, you have the formula of V0 is equal to Vmax into S upon Km plus S. Now, this says of S is equal to 100 km. So let us take S is equal to 100 km. So you have V0 is equal to Vmax into 100 km. Then you have upon km plus your 100 km, right? So you have V0 is equal to your Vmax, right? And this has 100 km upon 101 km, right? So this km will get cancelled. So you have a ratio of 101 upon 100, right? So I have this. So at 100 km, right, it is 101 upon what you call as your 100. So what fraction? So it is 101 upon 100. The next one you have here, P by O ratio of NADH and FADH2. So if you want to find out the ratio of NADH and FADH2, the P by O ratio, you know P by O ratio, that how many, like to simplify, maybe if I get some more time in the evening, late evening today, as we join every day at 9, that time we may discuss, but or we have so many things to discuss. Actually, in 2 days there are exams. But then P by O, you can say, is the amount of ATP formed per oxygen consumed. So maybe with one NADH, how many ATP you form, and with one FADH2, how many ATP you form. That is your P by O ratio. So anyone can tell एक NADH में से आपको कितने ATP मिलते हैं और एक FADH2 में से आपको कितने ATP मिलते हैं. Anyone knows there? 2.5 and 1.5. Great. So P by O ratio for NADH and FADH2 is 2.5 and 1.5. There you are, Ross. Thank you. The other people there, I hope it is clear how much ATP you get. Earlier, people used to believe that it was a integral value, but now we are sure, at least up to this point, that is 2.5 and 1.5. Reduction of 6 molecules of CO2 in the Calvin cycle. So when you reduce 6 molecules of CO2, you know you form 1 molecule of your glucose, because C6H12O6. So forming 1 molecule of glucose, you are going to require 6 molecules of carbon dioxide. So 6 molecules of your CO2. Right, so you want 6 molecules of your CO2 to be reduced, and out of these 6 molecules of CO2, what you are going to require, the reducing power which you require is both in the form of ATP and NADPH. So you know, for one CO2, जितना आपको पता है, एक CO2 के लिए आपको कितने ATP लगते हैं? 3 ATP और 2 NADPH. So one CO2 is 3 ATP and 2 NADPH. So 6 CO2 will be multiplied by 6. So you have 18 ATP. According to my calculation, you have 18 ATP and your 12 NADPH. I hope I am correct. So for fixation of 6 molecules of carbon dioxide, you will require 18 molecules of your ATP and your 12 molecules of your NADPH2. This is what we require for reduction of the 6 molecules of CO2. Coming to the next one now, reduction of atmospheric nitrogen. When you are reducing atmospheric nitrogen to form your ammonia, right? That is your biological nitrogen fixation, which takes place at normal temperatures and pressures. That is done by bacteria. And what does bacteria require? Bacteria require, it says, 8 electrons. It requires 16 ATP. it requires both of them, and it requires no ATP. Oh my god, अब ये normal temperature pressures पे तो ATP use करके ही करेगा, right? So it is going to use 16 ATP, and since it is reduction, it is going to require electrons also. So reduction of your nitrogen, it is going to require electrons, is going to require ATP, and hence your answer is both A and B. So you're going to require both A and B. That is your 8 electrons and your 16 ATP, right? So both will be required for reducing this what is called as your atmospheric nitrogen. And you know that they do this by the enzyme called as nitrogenase, right? And we know that nitrogenase is sensitive to something. Can you tell me nitrogenase is sensitive to what? Yes, Ross, it is both. Can you tell me nitrogenase is sensitive to what? Anyone can tell nitrogenase is sensitive to what? क्या आने के बाद nitrogenase अपना activity बंद कर देता है? Yes, great. High oxygen concentration. And you know that leguminous plants, they produce a compound called as DASH, which scavenges the oxygen from there. Like, which is that compound with the leguminous plants they produce? Anyone out there? Yeah, that's what I was also wondering, that why you said high, but then yes, it doesn't like oxygen, which is that compound now, which what you call is actually prevents it from what you call as oxygen, or scavenges the oxygen in leguminous plants. Anyone knows that? I'm just taking the mobile to look at your comments because अभी वहां पे लगा हुआ है ना. Yes. Which is that compound that scavenges the oxygen? Oxygen नहीं चलता ना nitrogen fixation ला, चालत नहीं, मग ते कोण घेऊन जातं? It is taken away by your leg hemoglobin, and leg hemoglobin is a protein which is going to scavenge that. And your cyanobacteria, the nostoc and anobina, which are fixing the atmospheric nitrogen, they also have some special cells where this atmospheric nitrogen fixation takes place, right? Yes, great, Ross, it is leg hemoglobin. Which are those special cells in cyanobacteria where nitrogen fixation takes place? What are those special cells called as? Do you know? Those special cells. Yes, great. That is heterocyst. Anyways, the phenomenon where the end product inhibits the synthesis of first enzyme at the DNA level, मतलब you have stopped the synthesis of the enzyme as such. मतलब आपने एंजाइम को बनाना ही बंद कर दिया, तो वो कहां पे हो रहा है? फीडबैक इनहिबिशन, फीडबैक रिप्रेशन, competitive inhibition, or allosteric inhibition. Please comment A, B, C, or D. The phenomenon where the end product stops the synthesis of the enzyme. Does not stop the enzyme there, right? It stops the synthesis of the enzyme. A, B, C, D. Just comment. Yes, Priyanka says feedback repression. You are right. It is feedback repression, right? Can you tell me, like, this feedback repression works with like lactose metabolism or tryptophan synthesis? You can just comment as lac, L for lactose, and T for tryptophan. If you think feedback repression is working for lactose metabolism, you can comment L. If you think it is working for tryptophan synthesis, you can comment T. So feedback repression is working for what? It is working for lactose, or it is working for tryptophan? Yes. As I told, at least comment your names and cities. I don't know why. I'm not going to come to your house. But tell me, right? Yes. Yes, T, it is. Yes, Manasvi says T. Yes, great. Priyanka also says T. It is the synthesis of your tryptophan. Anyways, all are polysaccharides in. Which of these groups contains all polysaccharides? You have sucrose, glucose, and fructose. Sucrose seems to be a disaccharide. D seems to be a monosaccharide. Gaya. Then you have maltose, a disaccharide. Lactose, again a disaccharide. Fructose, a monosaccharide. एक भी नहीं इसमें तो. Glycogen, a polysaccharide. Cellulose, a polysaccharide. Sucrose, oh no, gone kar diya. This is a disaccharide. So only option remained here is your glycogen, cellulose and starch. All three are polysaccharides. So cellulose, starch, glycogen, all three are polysaccharides. So can you tell me what are they made of? What is cellulose, starch and glycogen made of? Cellulose, starch and glycogen, all three are made of what? They're made of only one component, right? And what is that? I'm waiting for your answers. I know there is some gap in between like what I say and what you hear and then what you type. I'm keeping that much patience. Yes, it is D, glucose. I'll correct you, Ross, here. It is not always beta because I think in starch and glycogen you will have alpha. In cellulose you have beta, if I'm not wrong. What do you think? Yes, it is glucose only. Great, Manasvi. It is glucose. As far as cellulose is concerned, we can say it's beta. And as far as glycogen and starch, it is alpha. Am I right, Ross? What do you think? Yes, starch has alpha. I somehow think glycogen also has alpha because see, it's like they have that beta conformation and therefore that cellulose is like difficult to degrade. Whereas I think glycogen and starch will have alpha conformations and therefore they are storage. Yes. Oh, yeah. Thank you so much. Then you have how many different types of gametes which are being produced by this AABBCC. So how many different types of gametes will be produced by this capital A, capital A, capital B, capital C and small c? How many different types? Yes. Options are there. You have 2, 3, 4 and 8. How many different types are produced by this AABB and CC? I'm having a small board, man. Anyways, my heart is big. So which are these, right? Ross says 4. Yes. Anyone else with something else here? Let me use this a bit. I think we are done with this, so let me rub this so I can use this area at least. Oh my god. Board is not that good. I'll get new one soon. Yes. Ross says 4. Anyone else with different types of gametes for AABBCC? If I could help you out, like it is AA, then it is BB, then it is CC. Oh, this is one capital, one small. So one type of gamete would be A, B, and C. There would be A, B, and small c. So this is the first type. This is the second type. Oh, Priyanka said 8. I want to know which is the third and the fourth at least. 8 to jane do. So ABC is 1. This ABC is second. Yeah, Ross, I think it's 2. Not, you cannot get a different combination now. Can you get? Or koi aa raha hai combination? There are 9 people who are watching, but they are not telling their names. So you have Priyanka, you have Ross, then we have Manasvi here, and one is my wife. Like Muskan is there, and so Muskan, Manasvi, Priyanka, and Ross. I don't know who other 5 people are. So please tell your name at least. ये 2 ही आ रहे हैं ना? और कोई आ रहा है आपको gamete? और कोई आ रहे हैं? Anyone else? Ross, can you get any other gamete? Here, Priyanka, and other guys also. I just wanted to know, Ross, how many different types of gametes you can produce from this. So you have an individual which has this set of genotype. So when you produce gametes, you're going to get one from each. So in dono A mein se you will get A, in dono B mein se you will get B, and in dono C mein se you can get one capital and one small, right? Like if I'm having like capital T, capital, it can produce only one type of gamete. If I am having capital T, small t, it can produce 2 types of gametes, right? If I am having capital Y, small y, capital R, small r, it can produce 4 types. Capital Y, capital R, small y, capital R, small y, small r, and then you have capital Y, small r. So this is what I am asking, ki how many types you can produce. So from this, I think you can produce only these two types, right? I hope you got it now. From this, you can get only these two types. From every set, you have to take one. So this is only one type, this is only one type, so only variation possible is at this. I hope you got this one now. Yes? 2. So I hope you got it. Yes. Okay. So two different types you get. You can have a like simple thing like 2 raised to wherever it is heterozygous. So this is heterozygous at only one position. So it is 2 raised to 1, which will give you 2. If probably this would have been AA, BB and CC. This was heterozygous at 2 positions. So this would have given you 2 raised to 2 is equal to 4. If this would have been capital A, small a, capital B, the heterozygous at 3 positions. So this would have been 8. So it is 2 raised to कितनी जगह पे वो heterozygotic है, that will give you the number of types of gametes which are produced by this, right? Okay. The next one, the variations in the siblings are not observed due to, like all are responsible for variation except one of these, like the siblings will look exactly to their parents except out of these: independent assortment, crossing over, linkage or mutation, which will not cause or which is not the cause of variations. You can comment the numbers also, A, B, C, D, which is not the cause of variations: independent assortment, crossing over, linkage and mutation. Independent assortment is A, crossing over is B, linkage is C, mutation is D. You can just comment as A, B, C, D. Don't go till Z. Yes, please. Variations in the siblings are not observed due to. Yes, please. What does not lead to variations? Oh, you're still thinking? See, independent assortment will give you a different combination, like yellow and red. So independent assortment की वजह से येलो के साथ रेड जा सकता है, येलो के साथ रिंकल्ड भी राउंड जा सकता है, सॉरी येलो के साथ रिंकल्ड भी जा सकता है. You will have a different combination. Mutation will also result in a change, and crossing over will also result in the change. Yes. So what will not, just a second. I'm so sorry for this background music. So what will not result in the change is your linkage. Linkage में क्या होता है कि the genes they are present on the same chromosome. So possibility is that they will go together. It's a possibility. So this linkage is actually responsible for the parental combination. It is not responsible for the change, right? So here the answer is your linkage. The simplest one here, the phagocytic cells of liver. Which of these are the phagocytic cells of liver? The Kupffer cell, the acinar cell, the Langerhans cells and hepatic cells. Very easy one. Shreyansh, ये तो आपको ही पता होगा. Which is the phagocytic cell for liver? बता दो श्रेयांश. Kupffer, acinar, Langerhans and hepatic. I again request all of you to please tell your name at least, please. Priyanka says A, Kupffer cells. Anyone else there? Phagocytic cells of liver. आपका ऐसा है कि बैक्टीरिया में कहाँ liver है? Yes, it is Kupffer cells. I want to know about other seven members. Anyway, so we are done with today's microbiology. Some intelligent questions here. Well, these are also intelligent questions. These are not dumb questions. But then you have, okay, I have a series with me, 240, and then here something is missing. Then I have 120, then I have 40, then I have 10, and then I have 2. What would be coming here? Yes, please. Yes, please. 9. Let me multiply it for you. Let me multiply this by 5. I get this. Let me multiply this by 4. I get this. So let me multiply this by 3. I get this. Now can you tell me? Yes, great. So I multiply this by 2. I get 240. And I multiply this by 1. I get 240. So the answer is 240. I have one more here. You have to find the odd one out here. Out of 49, then 25, then 16, and then 81, which does not suit the group. This is the last one for you. '81. क्यों 81 क्यों बेटा? I would like to know the reason why 81. I don't see a good reason for 81. I'm afraid it's not. Vivek says 16. Vivek is preparing for competitive exams, I suppose. Thank you, Vivek. So, yes, it's 16 because here you see this is for 7, this is for 5, and this is for 9, which are prime numbers, and this is your even odd, you can say. So, this is the even number. Only one is the square of even number. So, don't be evil, be even, and prepare yourself. I would just like to know only this much today. I have some like personal work coming up, but since I had assured that I'll be there, I'm again apologize. I was late for 5-10 minutes here and there, and I'm going away quickly. But let's see. I'm planning to conduct it again today at 9, 8:30 or 9, something like that. So if I get time, I'll be conducting. I'll be dropping in a message at say around 7:30 or so. Once I'm confirmed, I'm available. I just want to know that whether you can join in the evening at around 8 or 8:30 if I tell you half an hour before. So please tell me yes or no, and also whether this is helping. So it can be 2 yes. Or if it is not helping, आप क्यों live आ रहे हैं ऐसे भी आप बोल सकते हैं क्यों हमको bore कर रहे हैं. So that is also there. So please tell me whether you can join in the evening and whether it is really helping you all, right? And I really hope that you are studying, and also you can tell me some specific topics on which you want me to take more questions. Most of you are now connected to me, but those who are not connected, you can drop in a message to me on this number. It is 9823683317. You can drop in a message on this number so that you will be updated. Maybe I'm not going to take many live sessions. I'm doing this for the PG entrance examinations till they are coming. But yes, if you really want to know and understand some concept, I would be highly obliged to do that. So thank you so much. And I'll not tell again and again to comment your names and city because you don't do. Thank you so much. Bye-bye. What happened, Ross? We can take now. You are crying, so you don't need to take a session in the evening. Yes. Should we take session in the evening, right? We can conduct session in the evening. Yes or no? At least tell yes or no. Yes, please. We can conduct session in the evening. Many are already gone. I don't want to talk. Anyways, bye bye. Take care. We can conduct session in the evening. At least tell yes. So otherwise, because I have to come back like all my day stuff. I have my lectures and sessions, and then I have to conduct this online class. So it's helping you, and we can conduct in the evening. Am I audible? At least you'll put a yes for that. Maybe no. Thank you. Bye bye. Take care. All the best. I'll decide for myself whether I should conduct the session or not. Or maybe there is a delay in the timing. I'll wait for a moment, right, and then leave. So if I could get 1 or 2 years at least, so I can conduct the session in the evening. I hope you all understand English at least. Yes. Is it fine? No response. Oh, thank you so much. Now I could see so many yeses. Thank you. Yes, I'm going to post this video. That's fine. That's fine. I thought, yes, bye, baby. Thank you. Thank you. Bye bye. Take care. So let's meet in the evening then.